I'm a little different. I'm a geriatrician some of the time at UCSF, and I run an aging biology laboratory at the Buck Institute the rest of the time. So I love that Dom and Jeff were talking about treating Navy SEALs and Army Rangers. I treat your grandma. But we're all interested in nutritional effects on health and the mechanisms that mediate those things. So I'm gonna expand on some of the topics that Dom started to touch on about what we're learning about the new biology of ketone bodies and how that fits into the picture of mechanisms of nutritional interventions and what that means for aging and disease. I haven't written a book yet. I have no disclosures. And this is science. This is going to be interesting and I think directly relevant translational science, especially for those of you who are clinicians and are trying to figure out how to think about managing patients who are trying interventions like low-carb diets or ketogenic diets or even these exogenous ketones. So this is science. But I know you guys aren't afraid of that after the great talks this morning. There were lots of molecules and enzymes and pathways going on. So we're going to do some of that too. We're going to talk a little bit about ketone bodies and normal physiology, which we've already heard a lot about, and then focus on the new biology of signaling activities of ketone bodies, and talk a little bit about how this could fit into actions in health and disease. I'm going to echo what Dom said, although I actually am a doctor, that kind of doctor, but none of this is medical advice. Do not take medical advice from people wearing microphones on stages. But I hope that you'll learn something from this that will be useful for you. In terms of practical questions, like when you're thinking about nutrition interventions or even exercise interventions, it's absolutely fascinating to me that we don't know what the right dose of exercise is, and we don't even know how to measure it. And the same is true for nutritional interventions. So this may give you some thoughts on how you can quantify and define doses of lifestyle, nutrition, and exercise interventions. And how to think about which elements of these interventions might be helpful for you or your patients to meet certain goals, and where the science, and hopefully the translational science, is going to be headed in terms of testing specific effects of nutritional interventions on conditions in health and disease, and my own favorite, aging. So the first part, ketone bodies are signaling metabolites. And I've gotten the sense in the last two talks that this is now becoming common wisdom. I love that. So how did I get interested in ketone bodies? Well, just from the observation, so remember, I'm coming at this from the angle of a geriatrician who studies aging and aging physiology and mechanisms of aging. And it's been really interesting to me actually throughout the day today that I sort of exist in a parallel and very closely opposed universe to the obesity universe. If you change the word obesity for aging in most of the talks today, we talk about exactly the same stuff. We talk about nutritional interventions like fasting and dietary restriction. We talk about the same pathways. We talk about insulin and IGF and glucose signaling and fat metabolism. We're really talking exactly the same language, just with a slightly different emphasis. So that's where I came from this, the observation that the interventions that have been known for the longest time to improve health, and particularly aging and longevity, especially in the laboratory, things like exercise interventions, dietary restriction, fasting, and now carbohydrate restriction, all share in common the production of ketone bodies. Are ketone bodies just a bystander in these interventions, or do they actually have a mechanistic biological role? You've already had an introduction to what ketone bodies are. Beta-hydroxybutyrate is the major ketone body that's induced when you fast or you exercise. So most of the extra ketone bodies that you generate are beta-hydroxybutyrate. That's one reason I'm going to focus on beta-hydroxybutyrate. So beta-hydroxybutyrate ketone bodies, they're fasting fuel. They're the way that your body mobilizes the energy reserves in your adipose tissue so that your heart and your brain, your muscles can use it when there's not a lot of glucose around. We've known that for a long time, and that's extremely important. But maybe this is interesting too, that ketone bodies are also fasting signals. And I'll show you how they bind to proteins, inhibit enzymes, and do other things that regulate broad cellular processes like metabolism and inflammation, and that these signals may convey some of the biological effects of the conditions in which they're made, of fasting and dietary restriction and exercise. I want to tell you how I think about what a ketogenic diet is. I think it's really important to remember, as we heard in the last talk, that there is no the ketogenic diet. The ketogenic diet is, I think of it as part of a continuum of low carbohydrate diets, where as you begin to restrict carbohydrates, at some point you reach a threshold, probably mostly determined by insulin levels, where your liver starts to make ketone bodies. It's not a switch, but there's a threshold where that production starts, and then as you continue to restrict carbohydrates, you make more and more ketone bodies. But it is a kind of gradual dose-response curve. So I think of it as different parts of the same continuum. And that's true in sense of their biological effects too, and I'll show you an example of that in a moment. This is data from mice, just to illustrate the point that as you gradually restrict carbohydrates in the diet of a mouse, you can go pretty far actually. You can have a mouse eating only 15% of its calories from carbohydrates and still really not making meaningful ketone bodies any more than it would in a normal diet. But then you start to see an increase. And the increase is gradual and accelerates as you reduce the carbohydrates further and further to nothing. So there's not a switch threshold, but the ketone body production is very sensitive to relatively small amounts of carbohydrate. And you get the most in mouse studies from nothing at all. This is how I think about the mechanisms of what do ketogenic diets do. And you could actually put low carbohydrate diet at the top of this hierarchy. But fundamentally, ketogenic diet has a couple of key components. One of them is carbohydrate restriction. In order to reduce insulin levels to the point where you reach that threshold where your liver starts to make ketone bodies. That's the ketogenic state. And that's the second characteristic. And then the third characteristic are the ketone bodies themselves that are floating around. All of these are biologically active, and all of these have health effects. Walking down that chain, then, ketone bodies themselves, I think of their actions as separated into energy functions, as a fuel, as the fourth food group, as Dom said, being burned to generate energy in the tissues, and then signaling functions, this new area that we don't understand as well, but where beta-hydroxybutyrate, acetoacetate, and other ketone bodies have these effects on cellular processes, in addition to their energy functions. So that's how I think about the hierarchy of mechanisms. Trying to figure out if there's a phenotype that you observe on a ketogenic diet, what is the mechanism, is a really complicated question. And this is how I think about how you might go about trying to answer that. Understanding that it'll be different for different phenotypes, for different conditions, for different diseases. In some, carbohydrate restriction might be really important and insulin might be critical. In others, it may actually be a direct action of a ketone body. I do think that ketone bodies in some contexts at least have additive effects, or at least the ketogenic state has additive effects over just carbohydrate restriction. And I'm going to show you a bit of aging data as an example of that. So my group with Eric Verdin at the Gladstone, now at the Buck Institutes, along with a group at UC Davis led by John Ramsey, both just finished a mouse longevity study of low carbohydrate and ketogenic diets. And we both tried to use those two comparisons in the best controlled way we can. So they're exactly the same diets in every way that we could control, except that we lowered the carbohydrate content almost all the way, but not so far that the low carbohydrate diet produced any ketone bodies. And then we lowered it to nothing, and that was the ketogenic diet. So almost exactly the same, except for that last bit of carbohydrates and then production of ketone bodies. And the data showed that the ketogenic diets, in yellow on the left and in red on the right, improved survival of these mice. In John Ramsey's study, it was more towards the latter half of life. In my study, it was more in the midlife, surviving into old age. But they both improved survival. And they both improved survival a little bit more than just the carbohydrate restriction alone, which is just a suggestion that the ketogenic part of that last step of carbohydrate restriction, added something on top of the low carbohydrate. But just the carbohydrate restriction alone had important physiological effects, probably a lot of that related to insulin. But maybe there's something related to the ketone bodies too. How I got into this scientifically, working with Eric Verdin, was on the tail end of a study that discovered a new signaling activity of beta-hydroxybutyrate, that it inhibits a class of enzymes called deacetylases. So our gene expression is controlled in an important way by the proteins around which our DNA is wrapped. So our DNA is usually very tightly wrapped around these proteins. Those proteins called histones have tails, and those tails can get modified in various ways, which affect the interaction of the DNA with the protein around which it's wrapped. Acetylation is one of the most important modifications, and more acetylation or less acetylation has huge effects on gene expression. and there's a whole panoply of enzymes that control this very carefully, how much acetylation there is, where it is, and taking it on and or putting it on, taking it off. So it turned out that beta-hydroxybutyrate binds to and inhibits certain of the enzymes that regulate this acetylation, like a drug. And if we looked at this with purified enzymes, we could show a nice linear correspondence between more beta-hydroxybutyrate causing more inhibition of these deacetylases. And then to make a long story fairly short, we found that inhibiting deacetylase enzymes changed the histone acetylation at the promoters of certain genes, regulated their expression, and those genes included genes involved in the antioxidant defense response. Probably a lot of other genes too, in a lot of different contexts, but that was the one thread that we pulled out of this. And that was published about 5 years ago now. Histone acetylation and deacetylases have really interesting roles in regulating aging in the yeast and flies and worms that aging scientists like to study. But I wanted to also point out a couple of human examples of their roles in age-related conditions and diseases. That we know that deacetylases and histone acetylation is important in age-associated cognitive decline and also in age-associated heart failure, diastolic heart failure or pressure overload heart failure. So some interesting hints that this regulation of deacetylases could have implications for chronic diseases of aging. That's not the only way that beta-hydroxybutyrate regulates gene expression. Just last year, Yingming Zhao, who is a fantastic mass spectrometrist at the University of Chicago, who studies these little things that get attached to histone tails, found that one of the things that gets attached to histone tails is also beta-hydroxybutyrate. It directly binds to those histone proteins. And we have no idea what it actually does there, but all we know is it's there. And if you fast a mouse, and fasting turns on expression of a whole bunch of genes involved in fat metabolism, then you find this beta-hydroxybutyrylation at the promoters of those fat metabolism genes. And again, we have no idea what it's doing there. This is kind of cutting-edge topic for research, but probably has something to do with regulating gene expression. You've heard a bunch already about the inflammasome, so this is another signaling activity of beta-hydroxybutyrate. What is the inflammasome? It's this sort of signal integrator inside of immune cells, inside of macrophages and other immune cells, that helps that cell decide whether to become inflamed when it senses damage. So the cells sense damage, and maybe that's bits of DNA, it's bits of mitochondria, it's reactive oxygen species, maybe it's amyloid in the brain. And then the cell decides whether that's sufficient to turn on an inflammatory response. If it is, then the inflammasome assembles from a whole bunch of little tiny subunits into this gigantic complex, which then turns on the expression of inflammatory cytokines, especially IL-1 beta. One example of this is uric acid crystals activating inflammasomes and triggering the inflammation associated with gout. And Deep Dixit at Yale, as you already heard today, did some really interesting work over a couple of papers showing that beta-hydroxybutyrate inhibits, and beta-hydroxybutyrate itself inhibits the assembly and then activation of these inflammasomes, and thereby reduces inflammation in various states, including particularly looking at a rat model of gout. I know that he's interested now in more translational studies looking at ketone bodies in beta-hydroxybutyrate and gout in people. But this is not just about gout. Inflammation, of course, but the NLRP3 inflammasome specifically have been implicated in a variety of diseases of aging. I guess today I should call these diseases of obesity too, and all of which have potential applications for signaling activities of beta-hydroxybutyrate. Beta-hydroxybutyrate is also a receptor for a couple of cell surface, or sorry, a ligand for a couple of cell surface receptors. One of them is called HCAR2. It's a really interesting receptor which is expressed in lots of cells all around our body. Beta-hydroxybutyrate is probably the primary ligand for it. It does a bunch of different things. It's how beta-hydroxybutyrate reduces lipolysis from adipocytes. And I'll return to that in just a second, as maybe part of a kind of self-regulatory mechanism to reduce its own production. But this receptor also regulates inflammation and also regulates neuron function in different tissues. There's another receptor, FFAR3, which maybe is most interesting for probably mediating the reduced sympathetic tone that you see in fasting. So in certain neurons, beta-hydroxybutyrate inhibiting this receptor reduces sympathetic tone and then lowers heart rate in laboratory mice. FFAR3 does a bunch of other stuff related to metabolism inflammation, where the role of beta-hydroxybutyrate is a little bit more shaky, not clear if it's turning it on or turning it off. But this is a particularly interesting bit of data. And then just a couple of months ago, we have a new signaling activity of beta-hydroxybutyrate, a new protein which it binds to, this time a protein that stabilizes RNA molecules. So remember, DNA is transcribed to RNA, is translated to proteins. So the stability of RNA, how long it hangs around there, is an important regulator of gene expression too. So beta-hydroxybutyrate binds to this protein, HNRNP-A1, and thereby allows that protein to stabilize the transcript of another gene called OCT4, which is one of the genes that Shinya Yamanaka found to be important for turning cells into stem cells. It's a quiescence factor or a stemness factor. But the point of this study was that by turning on this stem cell factor, it reduced senescence, especially in endothelial cells and vascular cells. So the bit on the right here, dark spots are senescent cells along the aorta of a mouse, and showing that increasing levels of beta-hydroxybutyrate reduces that senescence. Senescent cells are cells that are damaged, not enough to kill themselves, but enough to shut down, and then turn on an inflammatory response. So this is yet another way that beta-hydroxybutyrate might regulate inflammation, particularly in an aging context. So you've already seen this nice figure showing this is just one half of how I think about the signaling functions of beta-hydroxybutyrate. These being direct activities where beta-hydroxybutyrate is really acting like a drug, directly binding to something and inhibiting its activity or activating it. There's another whole set of signaling activities which are indirect as I think of them. They're the result of metabolizing beta-hydroxybutyrate into energy through acetyl-CoA. And this are changes in acetyl-CoA abundance, changes in the NAD balance, and things like that. And I won't talk about those today. So putting it all together, there's a variety of ways that beta-hydroxybutyrate can regulate inflammation through the inflammasome, gene expression through RNA stability, through beta-hydroxybutyrylation, through deacetylase inhibition, and inflammation and metabolism, both through cell surface receptors, all of which have really interesting implications for metabolic disease, diseases of obesity, and aging. And I'm talking mostly about beta-hydroxybutyrate. I think at this point we probably know the most about signaling activities of beta-hydroxybutyrate, but as Dom alluded to, we're finding similar things for acetoacetate and even, as I learned from Dom, for acetone. So all of the ketone bodies seem to have some degree of direct signaling functions, acting like endogenous pharmaceuticals. So beta-hydroxybutyrate is fasting fuel, but is also a fasting signal. How does this fit into aging? Is this a strange thing? And I just wanted to tell you that this is actually a great example of what we find with a lot of metabolites in an aging context. This is the link. It's a great example of the link between metabolism and aging. So in the aging world, I'm going to show you some kind of cartoon versions of signaling pathways that might harken you all the way back to undergraduate biochemistry, where you think about how signals from the outside world get into a cell. So you can have channels that let molecules through. You can have receptors that molecules bind to. And then these go through amplification steps, kinase cascades, where one enzyme activates a whole bunch of others, or second messengers. You make a whole bunch of a small molecule, and then that small molecule activates a whole bunch of other things. But ways to amplify the signal to get eventually the effect you're looking for. In the case of pathways that modulate aging, these are usually various forms of cellular stress response, autophagy and protein quality and things like that. What's interesting about aging is that many of these second messenger molecules that amplify signals are boring old metabolites that we all learned about in undergrads that have just canonical, simple roles in helping us to use energy. So NAD, AMP, S-adenylmethionine, acetyl-CoA, the kind of fundamental currency of energy in the cell that gets burned in the TCA cycle, acetyl-CoA. But remember I mentioned acetylation on the tails of those histones around which DNA is wrapped. This is probably the best known example of how these little metabolic molecules have entire secret lives as signaling metabolites. So acetyl-CoA is a substrate for protein acetylation, which is a widespread and hugely important way of regulating protein function, including the proteins that control our gene expression. NAD is now maybe better known as a substrate for a class of enzymes called sirtuin deacetylases, along with being an electron carrier that helps us to process the energy released from glucose and the TCA cycle. So this is kind of standard now, I think. We're learning more and more that more and more metabolites have these sorts of signaling functions, and this is how metabolism and aging are linked, and maybe it's how metabolism and obesity are linked too. So if BHB, if beta-hydroxybutyrate is a really interesting molecule, could we put it into a pill, and would that give us some of the same biological effects of a ketogenic diet? So this is getting a little bit speculative, but I just wanted to give you some tools to know how to think about the things that patients might come to you with. So first of all, there is no ketogenic diet in a pill. Ketogenic diet is of course a very complicated intervention that does a whole bunch of things in the body, and I kind of laid out my outline for you of how I think about those mechanisms. So if you think about how to pillify that, you need to first figure out what's the actual active component in whatever condition or phenotype you're looking at. And then you can maybe figure out how to bypass the sledgehammer that is the carbohydrate restriction and ketogenic diet and design more precise therapies. But there is no one ketogenic diet in a pill that will work for everything. But at least for some of the things that ketogenic diets do, ketone bodies are probably important, and now we're starting to have some tools in which we can parse this out mechanistically. So ketogenic diets in a pill. So just to remind you, beta-hydroxybutyrate is think of it as the circulating, it's the ketone body that travels. It's the one that goes around your blood, gets taken up by tissues that need energy. It's converted to acetoacetate inside of cells, both as the last step before it's produced and the first step before it's burned for energy. And then the other ketone body is sometimes lumped in with acetone, which is a spontaneous breakdown product of acetoacetate and gives you that fruity breath. So could you just eat beta-hydroxybutyrate or acetoacetate? You could try. Well, I guess you can. I think we all got a packet for that. Not exactly, though. The obstacle is that these are small molecules that are organic acids. And they're small molecules that are food. So we'd be talking about eating them in meal-level quantities. And they're rapidly metabolized, so you'd wind up having to eat an awful lot of an organic acid or an awful lot of salt if you try to neutralize it as a salt. But this is one way that you can do that. You can create ketone salts with sodium or other ions to balance out the acid. The trick, though, is if you're talking about a meal-level quantity, so say 100 grams of beta-hydroxybutyrate, which is about 400 calories, that would be a small lunch, that would include about 20 grams of salt, because this is such a small molecule, and something about a quarter of the weight of that is the sodium. That's a lot of sodium for a small lunch. It's the equivalent sodium content of about 5 liters of normal saline. So probably you don't want to do that every day. And remember, I'm coming at this from the angle of aging and chronic disease and things that we might do for months or weeks and months and years. But this is not hopeless. So first of all, there are some very clever and sophisticated ways you can try to balance this out to reduce the sodium content, use other ions, and maybe mix in other things. So some very clever ways that you can modulate this a little bit. But the real hope, I think, comes from thinking about this in a signaling function. So yes, it's a nutrient, but it's also a drug, and that drug has effects on its own. And maybe that drug, and we don't know this yet, maybe that drug has outsized effects beyond the amount that you're actually burning for energy. So there's a couple ways to think about, from these signaling functions, how beta-hydroxybutyrate might either inhibit or potentiate its own actions. So we already know from binding to a particular receptor on adipocytes that beta-hydroxybutyrate inhibits the mobilization of lipids, and that helps to put a tamper, at least somewhat, on its own production. It's a bit of feedback inhibition. But there are also examples emerging from the study of these signaling activities which look more like feedback activation. If beta-hydroxybutyrate changes gene expression in a way that enhances fatty acid metabolism, it might actually promote its own synthesis or promote the synthesis of other beta-hydroxybutyrate from other lipid precursors, as long as you have lipid precursors around. So maybe this is a biochemical basis for this idea of mixing beta-hydroxybutyrate salts, perhaps, a little bit of that, with other dietary lipids, even long-chain fatty acids, to provide some substrate for BHB to use. We haven't done or I haven't seen the experiments to really show whether this is what's happening in our bodies, but this is one idea that maybe there's a chance that small amounts of ketone bodies could have larger effects on metabolism. So ketone salts are one strategy for exogenous ketones. Another are beta-hydroxybutyrate precursors, so molecules that are metabolized to beta-hydroxybutyrate in the body. One example of this is the alcohol precursor butanediol, which Dom mentioned briefly a little while ago. So this is metabolized. This looks just like beta-hydroxybutyrate, except it's an alcohol instead of an acid, and your liver turns it directly into beta-hydroxybutyrate using the same enzymes that you use to metabolize ethanol. So that's very nice and very clever. Another group of molecules I think about as precursors are these medium-chain triglycerides. So what's different about medium-chain fatty acids, medium-chain triglycerides? Two things. One is the way that they're transported from the gut to the liver, which is more direct than for long-chain fatty acids. But the more important one is how they're transported into mitochondria once they get to the liver. So for long-chain fatty acids, insulin inhibits that transport. And this is one reason why you don't make ketone bodies if there's a lot of insulin around. But medium-chain fatty acids use a different transporter into the mitochondria, which is not insulin sensitive. So they can enter the mitochondria, and then the mitochondria has to figure out what to do with them, and generally turns most of them into ketone bodies. So that's the biological basis for the ketogenicity, so to speak, of medium-chain triglycerides. You can also combine these things, and this is the world of ketone esters. So trying to get around the organic acid nature of beta-hydroxybutyrate or acetoacetate by linking that to other molecules, and so sort of shielding the acid part of it. So the most commonly studied ketone ester so far, developed by Richard Veach and Kieran Clark, and the only one that's, as far as I know, that is available to the public, is a combination of this alcohol precursor, butanediol, plus beta-hydroxybutyrate. And you can see you sort of flip the butanediol around and just dock them together. So that's an ester. You can make an ester with just about anything. So you can combine these in all sorts of interesting combinations. You also heard about combinations of acetoacetate and butanediol earlier today. All of these are ketone esters. So ketone esters, like ketogenic diet, is a class description. There's lots of different ways to make a ketone ester. I just think of a ketone ester as anything that has a ketone body, acetoacetate or beta-hydroxybutyrate, which is ester linked to something else in order to shield that organic acid. So the way that ketone esters work is they're really just payloads. They're delivery molecules. So you have enzymes called esterases all over your gut and in your blood that will cleave this molecule at its ester bond. So it's an ester just so that it's safer to eat a bunch of it, but then as soon as it gets into your body, it's split into its constituents. And so in the case of the Veach-Clark ester, those constituents are beta-hydroxybutyrate and butanediol, and then the butanediol will be turned into more beta-hydroxybutyrate in your liver. Now, one thing to keep in mind is that butanediol is an alcohol. And alcohol is an almost necessary component of one of these esters. So when you split the ester, you'll generate an acid, beta-hydroxybutyrate or acetoacetate, and then whatever you've hooked them to will be in the form of an alcohol. So your liver has to process that alcohol. It's not ethanol, but it's some form of an alcohol. And since we're, again, talking about meal-level quantities, it's not a lot of alcohol, but it's something. So these alcohols are metabolized by the same enzymes that we use to metabolize ethanol. And if you're talking about, say, 30 grams of a ketone ester, that's the equivalent of metabolizing about half of a drink. And 30 grams is, I think, what people have settled on as a reasonable single dose of a ketone ester. So as a single dose, depending on your metabolism, that's probably fine. It's definitely something to think about if you're doing dose after dose, especially over a long period of time. So I thought I'd briefly talk about what we know about these exogenous ketones, ways of delivering beta-hydroxybutyrate directly in both rodent and human studies. And the slides are available, so you can look at this in more detail on your own. Most of the studies in rodents have used this Veach-Clark ester, butanediol and beta-hydroxybutyrate, and show an interesting set of metabolic changes, as well as Dom summarized earlier, a variety of interesting anxiety and memory effects, including in Alzheimer's models. And I do some work on looking at the mechanism of ketogenic diets in Alzheimer's models too. In people, most of what we know is through the study of, for ketone esters anyway, is through the study of elite endurance athletes. There is a little bit of literature about medium chain triglycerides going through a couple of small clinical trials in patients with Alzheimer's disease, which was fairly promising in small phase 1 and 2 studies, but wound up with a not so promising larger phase 3 study that it should be published soon. But most of what we know about ketone esters is in the context of young people taking them just one at a time and for exercise performance. There's not much, if any, literature right now on the use of exogenous ketones in older adults and people with chronic diseases or for the treatment of diseases, except for a case study of using one of the ketone esters in a colleague of Richard Veach's who had Alzheimer's disease. But that's the only person that I know so far who's been published to take a ketone ester in the hope of treating a chronic disease. But these studies are underway, and I think we're going to start to see them pretty soon and then start to see more and more of them. So I know there's a lot of interest in studying exogenous ketones in neurodegenerative diseases, but also in heart failure and diabetes and other conditions. So what does all this complexity mean for how to think about ketone bodies and ketogenic diets? Just a couple of examples of how these signaling activities contribute to pleiotropic effects on phenotypes like obesity and metabolic disease. So for example, there are several mechanisms that we can guess through which beta-hydroxybutyrate might affect metabolic disease. I already mentioned a couple, like effects of beta-hydroxybutyrylation on the expression of genes involved in fat metabolism. We know that certain deacetylases have roles in regulating insulin sensitivity. HDAC3 in particular controls gluconeogenesis. We know that the NLRP3 inflammasome can regulate inflammation in adipocyte, in macrophages in adipose tissue, and we know that inflammation in adipose tissue is an important mediator of metabolic dysfunction. And then there are also potentially effects through some of these cell surface receptors on mobilization of fatty acids and again regulation of inflammation, especially in macrophages. So all of these mechanisms might be relevant in different degrees to effects of beta-hydroxybutyrate on obesity and metabolic disease. And similarly in the kidney, there are at least 3 mechanisms through which beta-hydroxybutyrate may be renal protective in different contexts. So our own study looking at expression of antioxidant genes from beta-hydroxybutyrate, an interesting study of beta-hydroxybutyrate regulating prostaglandin production, and maybe that's through one of those cell surface receptors, we're not sure. And then a very recent study of beta-hydroxybutyrate protecting the kidney in a rodent model of hypertensive kidney disease through the NLRP3 inflammasome. So inflammation, probably inflammation, and repair. All potential molecular mechanisms that could mediate renal protective effects of beta-hydroxybutyrate. So all of this complexity also means that the biology is complex and the effects on people are complex and maybe hard to predict. We heard a great example of that this morning in the relatively straightforward idea of how much your blood sugar goes up after you eat a food. And you can imagine predicting how different people will respond to ketone bodies. All the information I just gave you should make you think that that's going to be very complex and difficult to predict, because you're not just talking about their ability to metabolize these exogenous ketones or their ability to generate ketogenesis or on a ketogenic diet. You're also now wondering about the state of their deacetylase genes and their gene expression, their epigenetic state, and their NLRP3 and their inflammasome, how active their macrophages are, all of which are both genetic and epigenetic contributors. So unfortunately, this is the normal state for studying aging and metabolism, that there is a huge component of individual variation. And I just wanted to show you a bit of mouse data, since I'm mostly a mouse biologist, that was a real revelation for those of us who studied dietary restriction and aging. So dietary restriction, we've known for 80 or 90 years if you feed a rodent less. They usually live longer. It works in most strains, but these guys did the really cool experiment of taking a couple of strains of mice, interbreeding them in ways to generate some genetic diversity, not nearly as much as we have, but at least some, and then dietary restricting them all. It turns out that dietary restriction, and then looking at lifespan, dietary restriction extended the lifespan of some of them. These are the bars on the left side of each graph. But actually, in many strains, dietary restriction made them live shorter, actually shortened their lifespan. Huge genetic diversity. And you can just think of how much more complicated we are than mice. So precision medicine and really understanding the genetic and epigenetic basis of how a person is going to respond to these large-scale interventions is the future. And finally, thinking about these signaling activities might give us some tools to actually be able to quantify what is the dose of a diet or exercise that you need to achieve a certain end. So maybe if beta-hydroxybutyrate binds to proteins, maybe you can measure that. Maybe that's a good measure of exposure to beta-hydroxybutyrate, and maybe you could use that as a threshold for when someone has had enough. Or you could look at downstream biochemical effects, like activation of inflammasomes or expression of certain genes. But these might give us hard quantifiable things to be able to tailor treatments to individuals with a certain goal in mind. So what I'd like you to remember as you leave here into camp for the cocktail hour, that beta-hydroxybutyrate is a nutrient and is used for energy, but also has drug-like signaling activities. I want you to remember this hierarchy of ketogenic diet mechanisms I showed you, of how to think about what a ketogenic diet is actually doing to create a phenotype. I told you a little bit about a taxonomy of exogenous ketones so you can know how to think about what is a ketone salt, what is a ketone ester. And then finally, I think that BHB signals will help lead us towards new clinical applications, give us some ideas of what diseases or syndromes or clinical problems might be most amenable to treatment with ketogenic diet or ketone bodies, and help us to guide making those therapies precise and individualized. So thank you for your attention.
We do have time for questions. You can jump right in.
That was, excuse me, is this on? Yes. Really a beautiful story. And I'm just curious about the beta-hydroxybutyrate butyrylation of histones and probably other proteins. I don't know how widely it's been looked at, but obviously if that's happening in a dynamic way, there has to be a deacetylase. And we have the sirtuin family, we have the non-NAD dependent deacetylases. Has that been figured out yet?
No, it hasn't. So great question, and we should talk later about that. This is a very active area of study. Mm-hmm. So I mentioned acetylation and beta-hydroxybutyrylation. Actually, just about any metabolite you can think of that's hooked up to an acetyl-CoA at some point in its life in the cell, we now know binds to proteins and modifies proteins and affects function. And we know some of the enzymes that, and so anything that's important functionally, there'll be enzymes that take them on and enzymes that take them off. Sirtuins, we're learning, are a class of enzymes that seem to have adapted to removing many of these different modifications. We don't know if any of them remove beta-hydroxybutyrylation yet. We don't know what enzyme does that. We also don't always know what enzymes put these modifications on. In some cases, it seems like much of that is non-enzymatic, which can still be regulatory because it's a function of the energy state of a cell. If there's a lot of succinyl-CoA around or a lot of acetyl-CoA around, that says a lot about the state the cell is in. But we don't know if there is an enzyme that attaches beta-hydroxybutyrate specifically yet.
Well, that was going to be my second question, because my guess is it probably is non-enzymatic, because it's probably not connected to acetyl, to the CoA component rather, at least not in the nucleus.
Yeah, we actually don't know yet we don't know that yet either. What's the mechanism even for non-enzymatic attachment of beta-hydroxybutyrate? This is very kind of, it's incredible that simple questions like this are unknown and cutting edge in 2018.
Wonderful conclusion to an exciting session. David Ludwig from Boston. You and others in the session have emphasized that ketogenesis is a highly regulated biochemical pathway. And one jumps over biochemical regulation with caution, because there are many examples in which unexpected and, in some cases, catastrophic, especially long-term consequences could occur. So in the normal state, high ketones is associated with low carbohydrate, both intake and circulating levels of glucose. Just a very rare exceptions, I guess, very high amounts of MCT, could alter that. Giving exogenous ketones changes that. Now, it's true, as was emphasized earlier, I think by Dom or Jeff, that ketones can suppress gluconeogenesis, and so you don't get very high levels of both. But if you're eating a high carbohydrate diet with a fair amount of protein and you're not oxidizing it, that gluconeogenic substrate is going to build up in a state of high ketones. I've heard anecdotal reports of people taking ketones on a high carbohydrate diet and feeling like an overwhelming burst of energy, but almost adversely so, like they couldn't sit still. Could we be setting ourselves up for overtaxing metabolic pathways that have acute benefits but long-term adverse consequences?
I absolutely, and I'm glad you brought up the idea of having caution when you're bypassing normal metabolic regulation, and I absolutely echo that and agree with it. And I'm going to return to something I mentioned about how the signaling activities of beta-hydroxybutyrate might regulate its own synthesis. The idea that there could, for example, be feed-forward activation of ketogenesis is interesting for the idea of maybe small doses could have biological effects. It's also caution-raising for the idea that you might reach a threshold where an overdose becomes a real possibility and a dangerous possibility because you're doing this element, beta-hydroxybutyrate signaling and ketosis, in the absence of what normally is the rest of the regulatory network in the context of fasting or carbohydrate-restricted diet. So I think there is absolute reason to be cautious about how we try these things and use them and really try to stay on top of our skis, so to speak, in terms of how we're advising the public in using them too. I think that one of the, so clearly I'm a fan of the exogenous ketone idea from a biological and medical perspective. I think one of the needs in that field, besides testing exogenous ketones in the setting of older people with chronic diseases and maybe with not the perfect metabolism of a marathon runner, I think is also really carefully studying the effects of higher doses and the effects of longer term use, just to really know what we're dealing with. I absolutely agree.
Or when in doubt, we could also just stick with the ketogenic diet.
Absolutely, absolutely.
We'll take one final question.
Yeah, mine sort of comes off of that, but the much more simple question for people that have relatives, loved ones with early mild cognitive impairment. If you had a relative starting that, would you suggest a ketogenic diet for them? Is it too early to actually say that, or can we just start doing that to family members and see some results?
So I stay over my skis, and I'll tell you why specifically in the context of Alzheimer's disease or early stage dementia. But first, that this is an active area of clinical investigation. And for example, there is a randomized controlled trial underway, and I know one of the investigators at Wake Forest who is running this, of a ketogenic diet in early Alzheimer's disease. Mm-hmm. So I think in the context of clinical investigation, that's a great idea. Mm-hmm. In terms of would I suggest my grandmother do it? Yeah. I have a hard time doing that, in part because geriatricians are naturally very cautious people, because we know how easily older adults can be hurt with the best of intentions, and how fragile they can be in certain ways. So I really don't want my older adults losing weight. Yeah. I don't want them having GI symptoms that could even be a delirium trigger, besides affecting their weight maintenance and other things. So I would be very cautious about that, given that we don't really have great data to say that we know it's a good idea yet. But it's clearly a good enough idea to study. Yeah. And I hope that there's more clinical investigation along that route.
Okay, great, thanks.
Yeah.
How do you define older adults? What's your
So we could talk for an hour about geriatrics, and maybe we'll talk over cocktails about that. But so the older adults that I would worry more about, what's a good way to say that? If you have to put your medicines into a paper bag when you go see the doctor, that's probably a good sign. Definitely if you've lost track of the number of chronic diseases someone has diagnosed you with, definitely a good sign. If you need help doing things day to day, and that's sort of the bread and butter of a geriatric practice, that's definitely a good sign. But age is not a number. Age is about your function and about how you act.